Hello, everyone. Thank you so much for joining us today for our webinar. I'm really excited to have Jamie and Lily here with me hosting today's session. I am Amy and I will be moderating today's session. Before we begin, I just wanted to let you know a little bit more about how the webinar will be run. The webinar is being recorded and the slides and recording will be available to you within 3 business days. A short Q&A session will follow the presentation to answer any viewer submitted questions that we may have. During the webinar, you can submit questions using the webinar sidebar menu. Select the Questions tab, type in your question, then hit the Enter key on your keyboard. Remember, you can submit questions anytime throughout the presentation. And that concludes my very brief introduction today. Jamie, I will now pass the webinar off to you. You may begin your presentation.
Thank you, and thank you for that introduction. As mentioned, I'm Dr. Jamie Willem, and today, along with my colleague Lily Zafis, we will be presenting on considerations for chromatographic method development and validations in food, beverages, ingredients, and other related products. So this webinar is part 2 in a 2-part series on method development and validation. Part 1 focused on method validation and verification, while part 2 will focus on method development. If you missed part 1, you can find a recording online or request access to the slides after the webinar. However, today I will be doing a short introduction and a summary of part 1, and then discuss considerations when developing a new method. From there, I will pass the presentation over to Lily to discuss considerations for instrumentation choice and some case studies on method development. So why do we care about method development and validation? Well, I imagine if you're here listening to this presentation, you already know the answer to this question. However, method development is required for any sample type, such as a new matrix or analyte that cannot be analyzed using an existing validated method. Method development can also range from small changes, such as adapting sample preparation, to starting completely from scratch with entirely new methods. For example, you may have an existing method for the analysis of an analyte or compound of interest in a beverage. For example, say sucrose in a fruity, sugary drink. However, you've developed a new product, such as a chocolate and peanut butter-based cookie, and you want to analyze sucrose in this material as well. In this case, you may be able to adapt your current method with new sample cleanup and preparation steps, or you may find that your current method does not work for this new product, perhaps due to matrix interference or co-eluting other compounds. In this case, you may need to start with developing a brand new method. Another example would be you have a new analyte or material for which there are no compendial or commercial offerings for this compound. This could be due to the compound being a new, novel material, such as a new pharmaceutical, or looking for improved methodology, or perhaps you need a lower limit of detection. In this case, you may not have current methods you can adapt, but would need to start fresh. It is not unusual for method development to involve work on multiple approaches simultaneously before eventually choosing the best fit. Following method development, method validation is performed, and method validation demonstrates that an analytical procedure is suitable for its intended use. Reliability or lack of reliable data produced from an analytical method can have widespread impacts on health and safety. That is because these methods that are developed and used are important in a wide variety of different industries, including food and pharmaceuticals. And so data integrity is important for continued product integrity. So I'm just going to give a brief summary of our previous webinar on method validation and verification, as this is an important component to method development. And you can see part 1 for the full webinar. So method validation or verification is the process of establishing the required performance criteria of a new, updated, or newly implemented method. In this way, we can show that a method is performing as intended and is considered fit for purpose. Fit for purpose is a phrase you may often hear when considering method development and validation. When we talk about fit for purpose, basically we are talking about a method meeting the needs of those who use it. For example, you may need to run an assay where the exact concentration of an analyte is very critical. The difference of a couple ppm may be significant. In this case, you would want a method that can have very tight accuracy and precision, and you'd want to set your validation criteria to reflect that. For example, you may set accuracy requirements of 98 to 102% of the expected value. On the other hand, you may have a method that requires less tight criteria. For example, you may have an impurity for which you just need to know the general level or detect, non-detect at a specific limit. That is, the difference between 10 and 12 ppm isn't significant, but the difference between, say, 10 and 100 or 100 and 1,000 is. In this case, you don't need super tight acceptance criteria, as was listed in the previous example, as this can lead to unnecessary additional method development and unnecessary additional work in validation. And so you want to set your method and your criteria to be fit for the needs of your particular data. Another important distinction is the difference between method validation and verification. I won't go into too much detail here as this was covered previously, but generally speaking, method validation is a more robust process involving a greater number of criteria and is typically carried out over multiple days with multiple instruments and potentially even multiple laboratories as needed. Verification tends to be a simpler process used to demonstrate that an already validated method performs as intended. Method validation is typically used when you have developed a new method or made significant changes to a method. For example, a change in equipment, such as improvements in detection due to new detector technology, or adding in a new significantly different matrix type. For example, if you, as with my previous example, if you ran traditionally a fat-free product and now you want to look at a product that is very high in fat. Verifications are performed when you are implementing a method that is already validated, perhaps a method transfer from another lab, or implementing a compendial method with performance data, or making smaller changes to a method. Note that there is not necessarily a strict line between method validation and verification, and each project should be evaluated on a case-by-case basis for what is required, both internally and from a regulatory perspective. So why do we validate or verify our methods? Besides our own desire to have a working method, it is primarily due to regulations. Many regulatory bodies require method validation for use in release and related testing in order to prove the integrity of the data. These groups, such as the United States Pharmacopoeia, US FDA, and ISO, typically require method validation. And they are also good sources alongside other groups such as AOAC for information on method validation and verification, and are great places to start looking when planning your own developments and validations. So now you know why we validate, but if you don't have a method to begin with, where would you even start? So today I'm going to briefly walk you through the basics of how to approach method development, especially if you are new to this process. Specifically today, we will be focusing on chromatographic methods, but you can apply these principles to any type of analytical method development. So where do we start? Well, the first step is to determine what information exactly from the method that you will need. That is, what is the final output of the method? For example, will this be an assay for a major component and you require an exact quantifiable number? Is this a screening method for impurities where detect or non-detect results is enough? That is it a quantitative or qualitative method? Also to consider are factors such as what sort of concentrations do you expect to see, or what detection limit do you need? What is the range of analysis that you are looking at? The answers to these questions will help you define a number of factors about your method that help focus your development and help you select the appropriate technique. For example, if you are analyzing a major component, say sucrose, like my previous example, that is present in high amounts say 5 or 10% of the total product, then you don't need to worry about detection limits. And in this case, you can look at a less sensitive detector, which may be less expensive, easier to maintain, and easier to run. However, if you are looking for trace impurities, for example, in the ppm or parts per billion levels, you will want to focus more on more sensitive and selective approaches. Similarly, if you are looking for quantitative results, you will want to consider a number of other factors, such as the required linear range, that is, how much variability do you expect to see in routine sample analysis, whether or not you need a calibration curve versus a single point, and any additional suitability you want to consider, such as check standards, required accuracy, and precision of the method. So once you have solidified what the output is that you need from the method, that is what the final data that you require is, the next step is to consider the analyte or analytes of interest. And this will help you focus your method development approach. And it will help you determine which approaches are even viable. For example, if your compound is a volatile compound, take for instance residual solvents such as hexane, chloroform, and related compounds, then you could look to techniques such as gas chromatography, which looks to analyze compounds that are already volatile, versus a non-volatile compound, say caffeine, which would be more readily analyzed using a technique such as liquid chromatography. Additionally, the structure of a compound you're analyzing affects detectors, which detectors you can use, and their sensitivity. So, for example, some compounds are very UV active, such as caffeine, while others give little to no response by UV, and therefore another approach is required. This is also a good place to consider sample matrix, as sample matrix can impact your sensitivity and selectivity. For example, maybe your matrix contains a lot of UV active compounds, and your analyte of interest is also UV active. These components may interfere with your analysis. In this case, you may consider a more targeted approach, such as mass spectrometry or fluorescence detection, or potentially looking to sample cleanup and preparation to remove unwanted matrix components to improve your method results. It is important to note that a lot of method development decisions are based on what is available. If you only have gas chromatography available to you, that is where you will start looking. If you're stuck in that situation and your analyte is non-volatile, for example, you may need to look into derivatization approaches. On the whole, there is often no one-size-fits-all approach to method development. The important factor is that you look for an approach that takes what you need from the method and what is available to you into consideration. That is, make your method fit for your purpose. So on this slide, I have a number of parameters that are important to consider when developing and running feasibility on a method. These are the same general categories you will see covered in method validation and verification. And so when you're developing your method, this is similar to what I talked to initially about what desired output is. You'll want to consider, for example, how selective your method needs to be, what kind of limit of detection, that is, how low do you need to be able to look for this compound, what kind of range and accuracy do you need. And it is important to look at each of these categories and to determine what is needed for the method you are working on. Part 1 of this webinar series goes into these in much greater depth. But if you are curious as to, okay, what's the typical accuracy required if I'm working on parts per million levels? There are a lot of great resources out there, such as the USP and AOAC, which can help you define these criteria if you're unfamiliar with working in this area. So now you have determined what kind of method you need. For example, an assay versus impurity test, quantitative versus qualitative. You've also taken the time to consider what type of method and instrumentation will potentially work for your analyte of interest. As well, you've established what sort of suitability parameters are important and what criteria you would need to hit to be able to make for routine use. Now at this point, how do you approach the actual process of developing the method if you have never done so before? Well, one of the best ways I have found to approach method development and look at these kinds of problems is to see what other people have already done. Some may refer to this as cheating, but others will call it standing on the shoulders of giants. But basically, you want to build off already established methods for similar analyzer matrices, and this is a great way to kind of hit the ground running with method development. So start by looking for methods that have already been validated. These may be methods already present in your lab that you're running on a regular basis, but you can also look to published methods such as those available USP, AOAC, and so on. You can find a validated method for a similar compound to what you're looking at. You'll often not be able to find the exact compound, otherwise you probably wouldn't be doing the method development. But if you can't find any validated methods, scientific literature is another great source. The scientific literature is typically on the cutting edge of method development, working with the newest novel compounds, matrices, and instrumentation. However, many of these methods do not establish performance criteria and are rarely validated. And so you may have to do some additional method development and feasibility on the front end. Finally, if neither of those approaches works, institutional knowledge is a great resource. Don't be afraid to talk to people more experienced than yourself or use past method development and experience to build off of. Once you've established a basic approach for how you're going to develop the method, the next step is feasibility. And feasibility should be used to determine both, one, does the method work? That's kind of the first important step. Up to this point, we've been mostly talking with theoreticals, but while you're considering all these factors, you should be running tests and seeing, can I detect this compound? Is this approach even feasible from the beginning? And then you can use a full feasibility study to determine matrix compatibility and additional refinement of the method. In addition, a feasibility study looking at detection range, calibration, linearity range, accuracy through spiking studies where you add a known amount of your compound to a product to see how much you can recover, how close to the actual value you can get, are critical. These feasibility studies give you some insight into how well your method performs and can help you select appropriate criteria for future validation. Often during this stage, you will find that you need to go back and reevaluate and redevelop your method if it is not meeting the criteria that you need. And this is an important and common part of method development. So do not be afraid to go back and rework where necessary. And often you'll find that you can run multiple method development approaches in parallel to try and determine the best approach and speed along development and feasibility. Finally, however, once you are happy with your validation or your feasibility, the next step is to write a validation protocol using the information gained from feasibility. It is important to write your protocol and establish your criteria prior to actually running the validation. In this way, you can show that the method works and meets your criteria, and you don't do it the opposite way where you wiggle your criteria to meet your method. And finally, the last step in method development is validation. The final piece of the puzzle is to validate your method and to perform the analysis as you outlined in your protocol and document the results to determine if they meet your required criteria. If you pass, congratulations, you have a validated method that is ready to be moved into routine use. However, if your method validation fails, don't throw away the data. Take a time to look through it and analyze what was wrong and use that knowledge to return to feasibility. Now I'm going to pass this presentation on to Lily, who's going to talk a bit about chromatography-related instrument platforms and case studies.
Thank you, Jamie. So now that we've gone over some initial considerations for method development, we're going to talk about chromatographic instrumentation options and how to choose a system that best suits your needs. So the first system that I'm going to talk about is a gas chromatography system with flame ionization detection, or GCFID. This type of detector is a good match for carbon-containing analytes, which are combusted in a hydrogen flame and produce ions that are then measured. Some of the best features of this type of system is that they are widely available and straightforward to run. They are also reliable and often sensitive with a wide linear range. This detector is generally considered universal for carbon-containing compounds, so this is a phrase you'll hear a lot for several different types of detectors. So no detector is truly universal, but we do make these broad statements if they respond to many different analytes. Some drawbacks to this type of system is that it can lack selectivity, so the FID will respond to any carbon-containing molecule in your sample, and that could include your solvents. Additionally, there are times where an FID does not have the sensitivity of a targeted detector, so this is analyte specific. So as an example, I have a chromatogram here to the right of a relatively pure analyte. You can see there aren't many extraneous peaks in this chromatogram besides the analyte, which is at about 8.5 minutes. And then, as I mentioned, FIDs do respond to many compounds, so that peak between 2 and 4 minutes is our large solvent peak. In this case, this was ethyl acetate. So our next system is gas chromatography with mass spectrometry. For this system, compounds are separated by GC and then ionized and identified by their mass. So MS can be run in tandem or single mode. And to expand on that, running an MS in single quadruple mode would involve scanning a selected mass range throughout your method. And the resulting chromatogram, which I have an example of here, is called a total ion chromatogram, or a TIC, which shows the total intensity of ions at any given time. So to take MS to the next level, you can then use tandem mass spec, which monitors a single ion and an ion that it breaks into. So this allows for a very high level of selectivity, which can filter out any interfering analytes. So these types of detectors are very sensitive typically, and they are also generally considered universal. So this definitely depends on the ionization properties of your specific analytes. The biggest benefit of an MS is the advanced level of selectivity that they can provide, like I've talked about, which can allow for compound identification. So as an example here, in this chromatogram I'm showing, this is the TIC of an essential oil sample. And if we just look at this one peak here, I have what just pops up on the right is the mass spectrum of that specific peak. So this shows the ions that were detected at that time. And then the two spectra that are shown below that are library matches from our library system, which the computer matched to the spectrum that was obtained during the analysis. So the computer then says that this compound, in this case, matches to bornanone, and it gives an estimate of the level of confidence for that identification as well. So when you're running GCMS, analytes will analyze and fragment in very specific and repeatable ways, and that's across systems and methods. So you can use these libraries to do identifications without any other information besides those ions they produce. However, with this system, it isn't only good things here, and sometimes the things that make this detector great are also the things that can make it not as great. So your sensitivity and the fact that you have universal detection can lead to more issues potentially with matrix interferences if you're trying to analyze a select molecule. And additionally, with this type of system, you are likely to need a more experienced analyst for development and maintenance. For our next system, we're moving into the liquid chromatography world. We're starting with probably the simplest and most common detector, which is a UV-Vis, or a photodiode array. This detector is good for compounds that contain chromophores, which absorb light at a specific wavelength. These detectors are known to be sensitive, and they are targeted since you can scan at a select wavelength that is highly responsive to your analytes. And in an ideal world, your matrix would not respond to that selected wavelength. If you're running with photodiode array, you can scan a range of wavelengths throughout your run, which gives you an absorbance spectrum that is specific to your analyte. So in this way, a PDAD detector can be highly selective and even used for identification. These detectors are also common and inexpensive and typically have a wide linear range as well. The only major drawback is that your analyte has to have a chromophore, otherwise you won't be able to see it with this detector. So the example chromatogram I'm showing on the right here, this is 4 overlaid UV-Vis channels for a chromatogram of a sunset yellow, or yellow 6 was our analyte, which is a common food dye. And I'm just showing here the pop-up is the UV-Vis spectrum for that peak. So in this method, we also did PDA scanning throughout, which we then used to confirm identification of this analyte if we see it in samples. Our next system is liquid chromatography with refractive index detection, or RI. In this system, the refractive index of a reference cell containing mobile phase is constantly compared to the refractive index of the mobile phase as it's passing through the column. So if an analyte is present, there will be a change in your RI signal, which is then measured. Perks of this detector is that it's generally universal, as most things will cause a change in refractive index, and these systems are also inexpensive and common. However, there are several drawbacks, including generally a low sensitivity and no selectivity, and a major one is that you cannot use gradients with this type of system, because if you had a gradient, then the refractive index of your mobile phase would be changing throughout the run as well. These detectors can also be sensitive to the environment, to things like temperature. RI detection is a lot more common in older methods, and it's not as widely used now in new developments because of some of these drawbacks. However, it's still the primary choice for some specific compounds such as sugars. So the chromatogram I'm showing here on the right is the sugar profile of an ice cream sample. So you can see fructose, glucose, and sucrose, and then additionally you can see a peak here at 3 minutes. And that peak is actually for water because we prepared our samples in water, but in this case our mobile phase was acetonitrile and water mixed. So that water injection had a different refractive index, and therefore we do have a peak there early in the chromatogram. Our next system is LC with evaporative light scattering detector, or ELSD. In this system, the mobile phase is nebulized and evaporated, and the analyte particles are passed through a beam of light, and then the amount of scattered light is measured. These detectors are often thought of as an alternative to RI because they are more sensitive and they are compatible with gradients. The drawbacks are that as a universal detector with increased sensitivity, you can have more issues with matrix interference potentially. And then in addition, since the solvent is being evaporated during these methods, non-volatile buffers cannot be used. These systems often tend to have a narrow linear range, so you might have to use a log-log linear curve fit. And then the chromatogram that I've shown here as an example, this is just one of the most common ELSD methods that we run in our lab, which is part of the monograph for taurine. The final system today that I'm going to talk about is liquid chromatography with mass spectrometry. So once again, this can be either single or tandem. And compared to GC, LCMS can use a variety of ionization techniques and solvent systems. So because of this, your identification may not always be as straightforward. So as an example, the primary ion you might expect for your analyte, instead of being something like M plus H, you might actually see an adduct with a mobile phase additive. So you might see M plus ammonia, or you might see M plus sodium if you have traces of sodium in your mobile phase. So it can be harder to predict how your analytes are going to behave because there's so many variables in this kind of system. So because of that, the systems with high resolution are necessary for identification, as these collect very accurate mass data, which can then be matched to known analytes. So drawbacks to the LCMS is that you can't use non-volatile buffers once again, and you can also have issues with matrix interference. So additionally, as with GCMS, it can be quite expensive and complex to maintain and develop methods on this system. So the chromatogram I'm showing here is from an LC tandem mass spec method. So that GC chromatogram I showed in the beginning for GCMS was a total ion chromatogram, and now we're looking at an MSMS chromatogram. So this method we developed for the analysis of capsaicin and dihydrocapsaicin, or DHC. And what I'm showing here off of the DHC peak is just the responses for the selected ion transitions that we were monitoring in this case. And I should note that our sample matrix in this situation was quite complex, but because we were using those MSMS capabilities, you can see that we had no noise in these channels. So we have very clean baselines, which meant that even in a complex matrix, we were able to have very low accurate detection. So now we're going to transition into some real-world case studies where we developed and validated methods with a variety of different goals and instrumentation. Our first case study is the analysis of a novel health molecule in a smoothie beverage formulation. So going back to what Jamie talked about, the first thing we did here is to consider what our goals for the analysis were. So in this case, we were looking at quantitation at the weight % level in a final product. So essentially, we wanted an assay for this final product. We also knew we were looking at a complex smoothie matrix, which would have many potential interference. And we were working with a novel molecule, so building off of existing methods or literature wasn't going to be as straightforward. So based on assessment of this molecule's structure, which was that it wasn't UV active, but it was organic and it was volatile, and with the goal of keeping this analysis cost-effective for future use, GCFID was selected as our platform for method development. So some of our feasibility studies first involved optimizing our instrument conditions to have good peak shape and our desired level of detection, which in this case was not particularly low because we were looking at assay levels. And then additionally, we did testing with different solvents for our sample extracts along with spiking studies to see if we could minimize the extraction of interference but maintain accuracy for our analytes. And we also tested multiple internal standards here. So the chromatogram to the right is just our final method chromatogram, and our internal standard is there at 4.6 minutes, and our analyte is at about 7.8 minutes. So the main challenges during this initial process was the complexity of our matrix, which we were expecting. So in addition to running final product precision samples, we used a blank smoothie matrix, which did not have the active ingredient to ensure that our accuracy study would be effective for our validation. And as I had said, we started this process out by using different organic solvents to try to reduce how much of those interference we were extracting. Our other main challenge was the novelty of this molecule, so we couldn't get commercial standards for this, and we did have to rely on a client-provided standard for our analysis. So to account for this, during our validation, we took our GC method and we ran it with mass spec detection, and then we used the fragmentation patterns of the analyte to confirm the structure for our specificity and to show that in our primary and secondary standards and in our sample that we had the same molecule. Our next case study is looking at analysis of a trace impurity in an artificial sweetener. So here we were looking at the opposite. We wanted to look at trace or low levels, which is going to be less than 1.1% in our final product. So we were looking for an impurity. Based on the structure and the expected low concentration, we selected LCMS for our method development platform. And this was also based on the decision that since we were looking at an impurity in what was going to be a large amount of a pure substance, by selecting a specific channel for monitoring by LCMS, we would be able to minimize the potential for issues due to the main components. So for feasibility, we optimized our instrument conditions, once again, to achieve retention and good peak shape. And in this case, we did quite a bit of optimization of our mass spec parameters as well, which I'll talk a little bit more about on the next slide. And we also did, as I said, some sample extraction testing to make sure that we weren't going to have interference from our main components. And then the chromatograms I'm showing here are just our impurity at lower levels, which is 0.02 ppm, along with 0.5 ppm. So our initial challenge during this development was achieving sensitivity that we needed. So our analyte ionized in negative ion mode. So if any of you are familiar with mass spec, you'll know that generally speaking, negative mode is much less sensitive than positive mode. So we did have to fight with our ionization conditions, such as our cone voltage and our other settings, in order to get ionization that would give us detection at the level that we needed. We also found that our analyte had more fluctuation in day-to-day signal than we had expected, which can happen with LCMS analysis depending on your analyte. So even though our sample results were consistent, we were seeing variations in our signal that ended up shifting our linear range between runs. So when we wrote our SOP and our validation criteria, we had to consider this and make sure that we were fitting the purpose of the analysis while still allowing for that flexibility in the ionization. Our next case study is 34 trace drug residues in wastewater. So for this method, we were shifting from that single component analysis that we've looked at, and we wanted to do a broader screening approach, but we still wanted to do quantitation and detection. And in this case, we were looking at extremely low levels in the ppb range. We also wanted to cover a range of drug residues, which had different structural classes. Based on the number of analytes and their variation in structure and their desired detection, we really only had one choice here, which was LCMSMS. Which is a very common platform for any type of broad screening method where you have many analytes. So the first part of our feasibility included identifying and then optimizing MSMS transitions for each analyte and making sure that the transitions that we selected had clean baselines to ensure that we would have low detection and high specificity. We also tested a variety of isotopically labeled internal standards for each drug class. So the chromatogram to the right is showing all of our analytes in our final method and the internal standards we selected. And these are all at 50 ppb in this injection. So you can notice that many of the analytes have a huge variety in signal intensity, even though they're all at the same concentration. So this is why we needed to make sure that our internal standard was matching and ionizing in the same way as the analytes, which is why we ended up having a different internal standard for each drug class we were analyzing. And then the final step here was actually a chromatographic method adjustment. So after we had our transition selected, we optimized our method to have the best peak shape and separation for as many analytes as possible. So with LCMSMS, you don't necessarily need separation, and sometimes you don't even want it. But in this case, it is ideal to have some separation so that your mass spec doesn't have to scan for too many transitions at one time. So the main challenge here, as I said, was method development for such a large number of analytes. So we had many different parameters that needed to be optimized individually. So this was quite an investment of time in the beginning to get the method going. Our other challenge in this method was determining our validation guidelines and suitability for this large number of analytes. So since our goal was very low level quantitation and detection, we relied heavily on EPA and FDA guidelines to ensure that we were meeting this purpose while still creating criteria that we were going to pass. So for example, a big part of our validation in this case was replicate sample spikes at a low level, which was 10 ppb. So to expand on that a little, in this case, our acceptance criteria had to have quite a wide range because this is an extremely low level to be analyzing. So for example, if our target is 10 ppb, but we have a recovery of 13 ppb, that's 130%, which in many cases wouldn't be accurate enough. However, in this case, when you're considering the difference between 10 or 13 ppb as a result, that really isn't a significant difference. So thinking about our criteria as we were writing this validation, it was significantly different than some of our other methods. Our final case study today is looking at the identification of 9 FD&C approved dyes. So the goal of this analysis was to qualitatively identify these food, drug, and cosmetic approved dyes, which is different than the other case studies we've looked at, which were all quantitative. And additionally, we wanted to leave and build in flexibility in this method and in the validation for future matrix extensions, because we wanted to have the ability to apply this method to any food or similar substance that we wanted. So based on our analytes being dye molecules with known absorbances, we selected LCUV as our platform. So for feasibility, we optimized our chromatography to have clear separation and strong UV-Vis spectra for each analyte. And we did test extractions and purifications on a variety of foods with known ingredients. The challenges for this method was to build in that flexibility initially, which is different than when you have a select matrix you want to target. So we wanted to make sure that we would have the ability to extend this method in the future as needed. And then additionally, because our goals were different here, the guidelines and process for validation were also different. So once again, we were relying heavily on our EPA and our FDA guidelines to ensure that we were meeting our method purpose, but still having flexibility. So for example, a significant part of our validation was the use of blind spikes, which demonstrated the ability of the analyst and the method to correctly identify any dyes that were present in an unknown sample. So I just have two example chromatograms that we obtained during our feasibility for this method, and the top one is from a grape candy, which contained blue 2 and red 40 dyes. And then on the bottom, we did an extract of a ranch flavored corn chip, and we identified yellow 5, red 40, and blue 1. So just to recap what we've gone over today, validated analytical methods are essential to ensure accurate testing of products that have a significant impact on human health and safety, such as foods and pharmaceuticals and many others. So in order to successfully develop a method with the goal of validation, it's important to first consider the goals of the analysis and also the properties of the analyte in order to choose a platform that is likely to give good results. So if you consider all of these factors before beginning, and then you use that in tandem with literature and institutional knowledge and other published methods, this can give you a really good starting point to hopefully make the whole process smoother. So here are just some helpful references and resources that we use often during this process. And you will receive a copy of these slides, so you don't need to rush to write anything down here. So at this time, I'd like to thank you for attending our webinar, and we'd be happy to answer any questions that you may have.
Wonderful. Thank you so much for the presentation today, Jamie and Lily. There have been a few questions that have come into the chat, so I just want to kick off with a few of these that have come in. So the first question that we had come in is, what makes an analyte more or less challenging to develop for?
Well, thank you for that question. So when we're talking about difficulty for analyzing an analyte or a compound, a number of factors can play into kind of that difficulty. For example, having analytes or impurities or other compounds present in a sample that have a similar structure can make it challenging from a selectivity and specificity point of view. It can make separation difficult. Other factors include, if you need a really sensitive method and your compound, for example, maybe doesn't ionize well by mass spec, or it doesn't have UV chromophore. But another important part of difficulty is, for example, matrix interferences. Matrix can often have a really large impact on sensitivity and accuracy. And in these cases, you might have to look into significant sample cleanup or instrumentation that allows selectivity for these analytes.
Great, thank you. The next question that we have here is, during method development, what do you do if you find that your setup and instrumentation aren't working as expected for the analyte?
Sure, thank you for the question. So sometimes even when you prepare and do a bunch of research, your analytes don't always behave how we think that they should. So at this point, it's important to go back to the drawing board and think about exactly what isn't working at that time. So there isn't a great one-size-fits-all answer to this question. But for example, if you're having more issues with sample matrix than you expected, then in that case, your answer might be to try using tandem LCMS instead of full scan to increase your selectivity. Or maybe you find out your analyte isn't ionizing well using GCMS, so you could switch to FID and see if you get better signal. Or even if you thought liquid chromatography was going to be the way to go, but it just isn't working out, you can look into derivatization procedures to get some volatility. So it's really important to think about exactly what your issue is, what isn't working, and then go from there and kind of go back to the drawing board.
Wonderful. Thank you so much. And then another one that we have that has come in the chat here. When analyzing multiple analytes in one sample, the example, the wastewater case study, is a standard curve being prepared each time the method is run?
In this case, when we did our drug screen, yes, we did have a standard curve for every analyte every time that we ran. This isn't necessarily necessary at all times, but in our case, especially for an LCMSMS method, because sometimes you see changes in ionization and response, from time to time, then it is important to make sure you recalibrate every time so that you are always getting, especially for trace levels, you know, in the ppb levels, you want to ensure you're getting as accurate results as possible.
Wonderful. Thank you for that insight. A couple more questions here. If you have access to a mass spec, wouldn't you default to using that for everything since you can't beat that level of specificity and selectivity?
Well, I can jump in on that one. So as Lily talked about in her part of the presentation, there are benefits and drawbacks to all detectors, and it really comes down to kind of that fit for purpose for your method. So in some cases, yes, the additional selectivity and sensitivity are really beneficial. However, in other cases, that can lead to increased development and routine costs, and it can lead to increased challenges. Mass specs tend to be more expensive and require more knowledge and training to run. So in some cases, if you can use an FID or a UV method, this can be an easier and less expensive approach and give you just as good results.
Wonderful, thank you. And I'm just getting caught up here, and I think that there is one final question. What can you do if your method shows great results for your analyte by itself, but not when it is in the final product?
Sure. So, I mean, we touched on this a little bit with several of the other questions, but so things to think about in this situation would be you have sample cleanup options or method modification options. So you could think about doing a solid phase or liquid-liquid extraction and try to remove the matrix from your sample, or you can try to adjust your detection. So for example, you could change your wavelength for LCUV so that something would respond better to your analyte and less to your matrix. So if you have the option to change your detector properties, you might be able to avoid sample cleanup, but otherwise that would be the way to go to try to get better results in your actual product.
Wonderful. Thank you so much. And it looks like we are all caught up on the questions. So I think that will wrap up our Q&A session today. I want to remind everyone that a recording of the webinar, along with a copy of the slide deck, will be available to you within 3 business days. So I know a couple of questions have come in of when you will receive that. Look for that within those 3 business days. And again, I want to thank you so much, Jamie and Lily, for your expertise and the presentation today. And thanks everyone for attending. Have a great rest of your day.